Hi everybody. Welcome back. We're going to be talking about active transport now. And once we're done with active transport, we'll get inside the cell and look at the different structures in the cell. So active transport is another membrane transport process, but in this case we're using some form of energy. Primary active transport uses the energy of ATP. ATP is energy currency that all of our cells can use. To move molecules either against their concentration gradient or to move very large molecules into or out of the cell. A secondary active transport, which we're not going to talk about here but we'll talk about later, is the movement of molecules using ion gradients. So when ions move down their concentration gradient, you have another molecule just jumping on that ion gradient and just kind of taking a ride with it. So that's a form of active transport called secondary active transport. So the first active transport mechanism is called the ion pump. And an ion pump is going to be charged or activated by the binding of ATP. When ATP binds and is hydrolyzed, it releases the energy from that second and third phosphate, and that activates the pump, causing the pump to push molecules against their concentration gradient, either into or out of the cell, depending on which way they have to go. This is one of the most famous pumps that we have. So this is known as the sodium-potassium pump. Sodium is one of our most common extracellular cations. Potassium is a very common intracellular cation. Sodium is constantly moving into our cells, in our nerves and our muscles, to cause our nerves and muscles to fire so that we can move or so that we can have electrical impulses. Since sodium moves down its concentration gradient, we have to pump it back out against the concentration gradient. Sodium-potassium pump does this. And how it works is you have 3 sodium ions bind, then ATP binds and activates the pump. So it is, once it binds, the binding of ATP changes the shape of the pump, causing it to open to the extracellular region. Sodium is released, potassium can bind. ATP is then hydrolyzed or broken down into ADP plus the phosphate. That activation is what causes the potassium or the sodium-potassium pump to change again in configuration, opening to the inside of the cell. Potassium can no longer bind, so potassium is released. This opens up the room for sodium to bind again. Oh, and I do have a video for that, sorry. See if it plays. Look at that. I don't know why the first one didn't play. How weird, right?
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. 1 phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration, That is to say, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
All right. Another form of active transport is the movement of molecules into or out of the cell using a vesicle, so vesicular transport. And there are different types of vesicular transport. Two major types are endocytosis and exocytosis. And there are three forms of endocytosis. So endocytosis is taking something into the cell. It's entering, endocytosis, entering the cell. The three forms are phagocytosis, pinocytosis, and receptor-mediated endocytosis. In phagocytosis, you have a large molecule, something like maybe a bacterial cell, that's going to be brought in, or a protein that's going to be brought into the cell. The plasma membrane surrounds the entire structure and pulls it in. The plasma membrane then blebs off, so you actually lose some plasma membrane, and you form this vesicle inside of the cell. Typically, phagocytosis is thought of as cellular eating because you're taking in large molecules. There are only a select number of cells of the human body that can undergo phagocytosis. These include your white blood cells, known as macrophages, neutrophils, your B lymphocytes, and eosinophils to some extent. Then we have pinocytosis. Pinocytosis is also known as cellular drinking because you're taking a small droplet of fluid into the cell, and within that droplet of fluid you have these dissolved particles. So pinocytosis can be done by almost all of our cells. And again, you're surrounding some material, some fluid with some dissolved molecules inside. You're blebbing off that vesicle, and you now have a membrane-bound vesicle inside that contains whatever dissolved materials that were in there. And then we have receptor-mediated endocytosis. This is a very specialized form of endocytosis. And in this case, you have receptors that bind specific molecules. Probably the most famous molecule that can be bound is cholesterol. And so when the receptors bind their specific molecule, if it's cholesterol or whatever, then the entire plasma membrane blebs off. So it forms a vesicle and pulls that vesicle into the cell. And so this here would be then a clathrin coated, because you have these little clathrin molecules on this side that is inside of the cell, and you have the cholesterol here, or whatever molecule is here. This is how our body gets rid of the cholesterol that is taken in through eating. So you don't have cholesterol buildup, and you don't have the problems with blood vessels becoming clogged. So if somebody actually has high cholesterol due to a disease like hypercholesterolemia, these receptors don't function properly, or they are non-functional, and so they can't bind the cholesterol, and the cholesterol ends up staying in the blood instead of being able to be moved into the cell. Exocytosis then is the opposite process. In exocytosis, you have material moving from the inside of the cell in a vesicle form, and that vesicle binds to the plasma membrane. That vesicle is composed of plasma membrane, and when it binds, it will just become part of the plasma membrane and release its substance into the extracellular fluid. So exocytosis, material is exiting the cell. Again, I have another video. I'm not going to go through this video, but you can definitely click on this link and watch the video on your own. If you have any questions over either passive or active processes, please bring them to class, okay? So now we can get into the internal structures of the cell. And so we're going to be looking at first the cytoplasm, and we're going to look at the different organelles within the cytoplasm. And then we'll go into the nucleus. All right? This is a figure from your textbook showing pancreatic cells. It's really hard to identify individual pancreatic cells and all of their organelles because, well, these are small structures. And cells, I mean, this is a microscopy image, and so you can see the nucleus, and you can see the cell here. You can see these little vesicles in these cells, but you can't see the organelles. And so we're going to look at this picture, which is just a diagram, generic diagram, but you can see the different organelles within this cell. So this, we're going to talk about these different organelles and how they function and how they help maintain cellular function. So we have both membrane-bound and then we have non-membrane-bound organelles. We'll talk about the membrane-bound organelles first, and then we'll talk about the non-membrane-bound organelles and their basic functions. So organelles basically determine the cellular function of the cell. So if you have a cell that has a specific type of organelle in abundance, that cell is probably specialized in doing certain jobs. Hopefully that makes sense to you, but if not, definitely come to class with questions because that's what I'm here for. Here you have the endoplasmic reticulum. So you can see endoplasmic reticulum is composed or has two different types, smooth and rough. The difference in the endoplasmic reticulum is if it's rough, it is embedded with ribosomes. So this is a series of membrane-bound channels that move from the nucleus outwards towards our Golgi apparatus, which we'll talk about. So when proteins start being produced, so protein synthesis starts in the nucleus, the transcript moves out, can enter the rough ER, and then can be translated into the protein within the rough ER on those ribosomes. So one of the functions of the rough ER is to produce and modify proteins. And then we package those proteins up and send them to the Golgi. The smooth ER functions in synthesizing phospholipids and steroid hormones from cholesterol. It also helps in breaking down certain toxins. So you end up having a lot of smooth ER in areas like your liver, whereas you're going to have a lot of rough ER in areas where you need a lot of proteins produced. And so again, you see that rough ER here. The rough ER is going to move from the nucleus to the Golgi. The Golgi apparatus is another series of tubules that function in packaging proteins and processing those proteins and then shipping them out. So when I think of the Golgi, I always think of a post office, because the post office takes packages and then it will send them to the areas where they need to go. Some of the materials from the Golgi enter directly into the cell and become cellular organelles themselves. Think of like lysosomes or peroxisomes. Some of the organelles, or some of the vesicles that come off of the Golgi, are sent to the plasma membrane to become part of that plasma membrane. And some are going to be secreted outside and into the extracellular fluid. And so I wanted to show you this picture because I could not find a good picture on Google Images, which is where I get most of my pictures from, of lysosomes. Lysosomes are your small little organelles that contain digestive enzymes, and lysosomes have a very important function. They're basically your digestive system. So what they're going to do is they bind to other organelles or other structures that are damaged, and they help to degrade them. They also bind to the vesicles that are brought in via endocytosis. So if you take in some large material like a bacterial cell, the lysosome can bind and break down that bacterial cell so it can't do damage to the rest of the cells of your body. And then we have peroxisomes. So peroxisomes are membrane-bound organelles that contain a different type of enzyme, whereas lysosomes contain digestive enzymes. Peroxisomes contain oxidative enzymes. Oxidative enzymes then are used to break down toxins and via oxidation. And they also function in oxidizing free radicals that are found in your system. Peroxisomes can be used to break down fats through beta oxidation so that we can use them in processes like aerobic respiration. You're going to have a lot of peroxisomes in areas where you are constantly trying to detoxify materials, toxins. And so the liver, again, is a very important area for peroxisomes to be found. The next organelle is the mitochondria. Mitochondria, I think, in my opinion at least, is one of the most well-known organelles. It is the powerhouse of the cell. It has a double membrane. So you have this inner membrane. Right there's your inner membrane. And then you have an outer membrane. It kind of looks like a kidney bean with the inner membrane moving in and out, in and out. The inside of the mitochondria, the very inside, is known as the matrix. And then those little grooves, those are known as the cristae. The mitochondria's major function is to produce ATP energy, which we will talk about minimally this semester, but we will definitely get into it when we're in the digestive system, and we talk about metabolism next semester. And that's a really, really important process. So I think you're going to definitely get to know the mitochondria very well by the end of your, by the end of your A&P career. Then we have non-membrane bound organelles. We have the cytoskeleton, which is composed of 3 different proteins. We have microfilaments, intermediate filaments, and microtubules. Then we have centrioles, which make up your centrosomes, I'll say it that way, which are important in moving your DNA around during cell division. And then we have ribosomes. All right, so here are the cytoskeletal filaments. The cytoskeleton's major function is to maintain cell structure and integrity, but it also functions in movement of materials throughout the cell. So we're always moving proteins and other materials, vesicles, whatever else. And so we have the cytoskeleton inside the cell that helps in movement of those materials. Cytoskeleton also functions in movement of DNA. And so here we have, the entire thing is called a centrosome, but they're composed of centrioles, and centrioles are made up of microtubules. Microtubules are part of the cytoskeleton. So the centrosome is actually, in a nutshell, part of the cytoskeleton. And its major job is to connect to your DNA molecules during cell division so that we end up with a single DNA molecule of each type in each individual cell during cell division. Cilia and flagella are not actually organelles, but they are made up from the cytoskeleton again, or from cytoskeletal filaments. So I always talk about them here. These are actually extracellular structures that are found on certain cells. So they would be extensions of the plasma membrane. Cilia is smaller in length than flagella, and its main function is movement of materials along the surface of a cell. So over here you see the trachea. Trachea contains ciliated cells, and these ciliated cells beat mucus away from the lungs and towards the throat so that that mucus doesn't get trapped in our lungs, and any materials that we breathe in don't get into our lungs, and instead we can spit or swallow that material, and then it doesn't cause us breathing problems. And here's a prettier picture of the actual ciliated cells within the trachea. And so here you can see the actual cilia along those, and this is really beautiful because you can really see those individual cells within the lung tissue, or within the tracheal tissue, with the nuclei and everything. So I love this picture. I do, I'm not going to lie. All right, so flagella then are similar in structure to cilia, but they tend to have longer, so I think of cilia as kind of like finger-like projections, whereas flagella are like long hairs, so they're a lot longer. And in the human, the only flagellated cell is the sperm cell, and its job is to propel the cell towards some stimulus. And in this case, flagella are constantly moving towards the egg. That's their job, that's their goal, is to find that egg. And the last structure that I like to talk about is microvilli. Microvilli are very small extensions of the plasma membrane. They're very, very densely packed extensions, and their major job is to increase surface area for absorption. So we find a lot of microvilli in intestinal cells, as well as in our nephrons of our kidneys, where we have a lot of filtration and reabsorption occurring. All right, I'm going to stop this video because we have just finished the organelles, and now we're going to start on the nucleus, the, I guess, brain of the cell. Okay? So I'll see you in a little bit. Bye.